So outline for chapter 10 will be 10.1, 10.2, 10.3, and 10.6. So when I say the outline, this means that these topics, these subtopics are very important for you to understand, and also it will come out in your exam. Okay, those that are extras, meaning like I said today, I inserted a bit of cancer subtopics in here as well. It is just for your own knowledge. Okay. Right, so let's look at the learning outcome. Okay, the first one is to explain the stages and major events of the cell cycle. The second one, you will be able to list the checkpoints that regulate the progression of cell through the cell cycle. These are the learning outcome that you have to achieve by the end of this lecture. Okay, so by the end of the lecture in about 1 hour time, you should be able to achieve these 3 learning outcome. Okay, so the third one is summarize the stages and major events in mitosis and cytokinesis. Okay, so what is the essential code of life? Can anybody just tell me what do you understand the meaning of the essential code of life? Siapa-siapa? It's okay, in BM boleh, in English boleh. What do you actually understand? The essential code of life. What does it mean? What is it referring to? Anybody? Yeah, it's okay. I won't judge. You can just, we can have a two-way communication. What is
Our DNA code.
Yes, correct. Our DNA. Why is it called the essential code of life? Why do you think it is called like that?
Because our DNA contains everything who we are. How, like, for example, maybe the pigment of our skin, the color of our
Yes,
eyes, so
correct,
on and so forth.
correct. DNA contains the coding, okay, coding for the protein nanti. You will learn more on that. That is the essential necessity for our cells to coordinate. Faham tak? So this DNA have the major coding code for the body, for our apa ni? Our human body to survive. Contohnya coding for growth, coding for disease resistant, coding for animal development, or coding for anything. So all of these are in the DNA. That is why it is called the essential code of life. Okay? So DNA are in the chromosome, and chromosomes are in the cells, and cells, we have them in the organism. We are full of cells. Okay, so the first one, we'll go into the first subtopic, which is 9.1, which is the cell cycle. So if you see your meta textbook, please refer on page 148. Okay, so the cell cycle. What is the cell cycle exactly? It is a series of steps, okay? Series of steps that all cells go through prior to the initiating the cell division. Okay, these are the cell cycle. So just prior to the next division, the cells have to grow larger and the number of organelles doubles and the DNA is replicated. Okay, so there are two major stages of the cell cycle. We have the interphase. So if you see here, the interphase comprises of 3 main steps, which is G1 phase, which is before DNA replication. S phase, the synthesis phase, is the DNA replication. And the next one is the G2 phase. So G2 phase is after DNA replication. And then we have the mitotic phase, so M phase, we call it, okay? M phase stands for the mitotic phase. So this is where the mitosis and cytokinesis occurs. So these all adds up to the prophase, to the metaphase, anaphase, telophase, and then the cytokinesis. All of that are in the M phase. Okay, interphase have three major steps that you have to know, and then we have the M phase. Okay, so we'll go into interphase. So this part right here, the G1, S, and G2. So most of the cell cycle is spent in the interphase. Usually about 90% of the cells, 90% of the cell cycle are in the interphase cycle. So cell performs its usual function in the interphase cycle. Okay, usually dulu yang masa kita belajar, masa awal dulu saintis tak berapa nak do banyak research on, they call the interface as the resting phase. But they found out later that it's not. It's actually the busiest phase is the interface sebab dia ada banyak dalam tu, dia more to the replication and then the duplication. So that is why it is one of the busiest phase. It's called interphase. So the time spent in the interphase varies by cell type. Okay, ada yang cepat, ada yang can complete the interface in matter of a few hours je. Ada yang take a long time to finish the interface and then go on into the M phase. Okay, it depends on the location of the cells. Okay, so nerve and some muscle cells ni contoh macam heart muscle cells, okay, they do not complete the cell cycle, meaning they remain in the G0 stage. Pula dah ada G0 pula kan. So G0 ni is this part here where the cells go through a rest, they call it. Okay, dia tak complete, dia tak go on into replicating, dividing. Sorry, they are in a resting phase here, G0. Okay, so most of the somatic lah selalunya yang tak ada yang embryonic cell yang go through G0 stage, tak silap saya. It's only mostly somatic lah yang kalau yang that I know of. Usually nerve and heart cell, red blood cell, okay, that ada masuk dalam G0 phase. Okay, repeat again into detail with the G1 phase, the S phase, and the G2 phase. So G1 phase, the cell is in recovery from the previous division. And then these are the important 3 points that you have to remember happen in the G1 phase, which is it doubles its organelles, it grows in size, and it accumulates raw materials for DNA synthesis. Okay, sebelum dia masuk kepada S phase. Okay, dalam S phase pula, replication takes place. Okay, S, you have to remember S for synthesis. Okay, S, replication. So the protein associated with DNA are synthesized. And then the chromosomes, okay, let's say the first part, chromosomes enter the S phase with one chromatid each. Okay, so by the end of the S phase, they will have two identical chromatids, or sister chromatid we call it, each. Okay, so these sister chromatids remain attached until they are separated during mitosis. Okay, so that's what happened during S phase. It is synthesized from one chromatid, dia jadi two identical chromatids, or known as sister chromatids. Next, after S phase, we have the G2 phase. G2 phase is the phase after DNA replication. G2 phase. Okay, it is like to get ready for mitosis punya phase. Okay, it is located between DNA replication, which is the S phase, and the onset of mitosis. Okay, so here cell synthesizes protein that are necessary for cell division, such as the microtubules. Okay, microtubules ni apa? Will form lastly the spindle fibers. So this is the phase where it gets ready. G2 phase. Okay, G2 phase, S phase, and G1 phase are in the interphase part of the cell cycle. Interphase, as I said, comprise of 90% of the cell cycle. So the last 10% is from the mitotic phase. Right. So you can stop me anytime if you have any questions, okay? Sometimes I get carried away. So please stop me and ask. Okay. All right. So let's go in detail on the interface. This is what happened. So the chromatin become condensed, right? So these are the interface. And then the centrioles are formed, the centrosome. Centrioles ni lama-lama dia akan extend the microtubules and akan jadi the spindle fiber in prophase. Okay, this is the interface here. This part to the left is interface, and we have the mitosis, which is the M phase here. Contoh, okay, this is what happened. Dua ni kalau you compare, this is what happened in interphase. Okay, it gets the cell ready for the cell division. Okay, kalau you perasan dekat sini, okay, just nanti we go into detail. Tapi if you perasan, gambar ni is good to see the centrals. Okay, now here, this is the interphase, and in the prophase, dia jadi early mitotic spindle. This is kalau mitosis. Kalau meiosis, it is meiotic spindle. Okay, mitotic spindle, dia akan go to the post, baru akan ada prometaphase. Prometaphase ni dia tak align lagi. Only the spindle fibers are connected to the kinetochore. Dia belum align. Dia baru lekat je. So that is the prometaphase. Okay. Alright. So next, balik ke tadi, after interphase, we have the M stage, which is the mitotic stage. Okay, tadi kan kita dah ni from G1 tadi round, pergi S phase, and then kita round balik ke G2, and then now is the mitotic phase. So mitotic phase ni ada dua that you have to remember. It is mitosis, and the last one is the cytokinesis. Mitosis is nuclear division. Okay, cytokinesis is the division of the cytoplasm. Don't be confused. Cytokinesis is not nuclear division. Okay, last time ada juga yang jawab masa dalam exam, mitosis dia tulis cytokinesis dia tulis nuclear division. That is wrong. Okay, cytokinesis is the division of the cytoplasm. Mitosis is the nuclear division. So in mitosis, there are many phases. There are prophase, there are prometaphase, there is metaphase, there is anaphase, and there's lastly the telophase. Okay, and in mitosis, daughter chromosomes are distributed by the mitotic spindle to two daughter nuclei. Okay, so after mitosis and cytokinesis, it results in two genetically identical daughter cells. Okay, we will go into detail in that. Okay, so what controls the cell cycle? Okay, dia mesti ada signal, betul tak? Okay, a signal is a molecule that influences the activities of a cell. So signal ni ada dua that you have to remember. It is either internal and also the external signals. Okay. Internal signals contohnya yang selalu yang kita fokus on are cyclin. It is here. This cyclin is a family of protein yang akan increase and decrease as the cell cycle continue. Dia whether kalau too much cyclin, dia akan kurangkan. Dia sendiri yang adjust. So these are called the internal signals. Okay. So specific cyclins must be present for the cell to proceed from one stage to the next. Okay. So internal signals that we focus on in our syllabus are cyclin. Okay. External signal pula can be other, banyak. So now we concentrate only on growth factor signaling protein, which is received at the plasma membrane. Okay, nanti we'll go into that juga lagi. So what happens if there are no signals atau no internal signals? So without cycling, the cell cycle will stop at the checkpoint. Okay, tadi kita tengok ada banyak phases kan? Banyak phase G1, S, G2. Okay, between one phase to the next tu, kita ada checkpoint. Dia akan check dulu sama ada DNA kita tu okay ke tak, it has doubled ke properly or not, atau ada any damage to it, then dia akan stop. Okay, so without cycling, this family of protein ni, dia akan stop at G1, M or G2. Okay. So damage by solar radiation or X-rays are repaired at the G2 checkpoint. Okay, let's go into detail on checkpoints. So what are checkpoints? It's like a roadblock. You lepas ke, you tak lepas ke. Okay, so it is a point where it is macam quality control punya checkpoint. So this is where the potential points along the cell cycle during which the condition of the cells are accessed. Okay, they are accessed. Okay, mana gambar tu? Okay, if you refer to figure 9.1 in your media textbook, you can see ada macam gambar stop sign. Tapi stop sign tu dia tulis G1, G2 and M. Okay, do you guys see that on page 148? Okay, those are checkpoints. Okay, checkpoint G1, is between G1 to the S phase. Kalau checkpoint G2 is after the S phase, from G2 nak masuk ke mitosis. Okay, that is the G2 phase. Okay, the last one yang M checkpoint ni, kalau you tengok refer to your cell cycle dalam media, M point is semasa mitosis tu, usually masa metaphase. Okay, that is the M checkpoint. Okay, so depending on the signals received and the condition, the cycle will either stop or continue. Okay, kalau dia rosak, damage ke apa-apa, dia akan stop unless there is a chance to repair. Kalau it is all okay, is good to go, then dia akan lepas the checkpoint. Okay, so blocks cell cycle progress unless specific molecular and physical conditions are satisfied. This is what I meant by dia akan try to repair. Kalau tak boleh repair sangat, dia punya sel tu memang dah terlalu teruk atau dah mutated teruk, tak boleh nak repair langsung, it will go through a self-destruct mechanism. We call it as the apoptosis. Okay? Okay, so this involves signaling protein, cycling, which is internal factor, and other hormone or growth factor for external signal. So the importance is to control the mechanism in the cell to ensure proper division of the cell. So that is why we need to have checkpoints. Kalau tak, dia akan out of control. So kalau out of control, it could lead to tumors and also cancers. Okay. Alright. So this is what this is the figure 9.1, one I was telling you about. Okay, G1 checkpoint again. This is the primary checkpoint. Okay, G1 ni sebelum dia masuk nak replicate, dia kena check dulu. These are okay ke tak? Kalau DNA ni damage, apoptosis will happen. Okay, apoptosis ni is self-destruct atau program cell death. The cell progresses through a series of events where it is engulfed lastly by the white blood cell. Okay. So this is G1 is the cell cycle main checkpoint. So if DNA is damaged, like I said, apoptosis will occur. Kalau dia tak boleh nak repair lah. Kalau dia boleh repair, it will continue. Kalau dia tak boleh nak repair, it will go through apoptosis. Kalau cell too good to go, then the cell will divide when the growth signals are present and nutrients are available. Baru dia masuk kepada next phase, which is the S phase for growth and replication. Okay, that is the first checkpoint. Jadi 3 checkpoint altogether. So the next checkpoint is the G2 checkpoint. Okay. Okay, dalam your exam, kalau dia suruh list ke apa state ke the main the checkpoints in the cell cycle, jangan tulis primary checkpoint. Write G1 checkpoint. Okay? G1 checkpoint, G2 checkpoint, and also M checkpoint. Jangan tulis primary checkpoint, jangan tulis mitosis checkpoint, and don't write spindle assembly checkpoint. That is only for your reference. Okay, G2 checkpoint. So dia akan prepare for mitosis between G2 and M. Ada G2 checkpoint. So what happened here? Mitosis will occur if DNA has replicated properly. Kalau dia tak juga, dia tak boleh juga mutation happen ke apa ke, ataupun dia dah salah replicate ke dekat sini, apoptosis will occur. Okay, if the DNA is damaged and cannot be repaired. Alright, so if here DNA is damaged, apoptosis will occur. Dekat G2 kat sini, damage and cannot be repaired, then apoptosis will occur. Okay, so the last checkpoint is the M checkpoint. M checkpoint ni is the spindle assembly checkpoint. What does it mean by spindle assembly? Okay, masa M metaphase phase ni, dia apa, all the chromosomes will align on the plate or the equator. Kalau dia tak align properly, then it doesn't pass the M checkpoint. Okay? So mitosis will not continue if chromosomes are not properly aligned. Kenapa kita nak kena di-align? So that we know that it is correct, the chromosomes will be correctly distributed. Okay, if this part here salah, then apa ni, non-disjunction can occur, and also apa ni, banyak lagi all this apa, what they call it, all the mishaps lah happen. So kita tak nak the chromosome are not equally distributed between the daughter cells. Okay, so because that will lead to other types of mutation lagi. So that is why we need to have a M checkpoint here, which is the spindle assembly checkpoint. Okay, so that is all for the 3 checkpoints. First checkpoint is very crucial, which is the main checkpoint. And then we have another checkpoint before kita masuk kepada mitosis, and then the last one before dia continue mitosis, continue after metaphase. Okay. Right. Okay. Another condition yang ni, G1 checkpoint. G1 checkpoint yang tadi saya mention tu, dia kena make commitment to divide by evaluating the growth signal, determine the availability of nutrients, and also access the DNA integrity. DNA integrity ni maksudnya dia kena check the DNA which are free of errors and also are not damaged. What happen if it's damaged? Apoptosis will occur. Okay, it will destroy. Atau dia kena, nanti dia akan makan dengan the white blood cell. Okay, does apoptosis happen all the time in the cell cycle? It is there, but it doesn't happen all the time. Only when there is a damage in the DNA or the DNA cannot be repaired, then it will go through apoptosis. It doesn't happen every single time. Okay, macam cell division tak. Okay, apoptosis is there, cuma it doesn't, kalau tak ada DNA damage dan tak adalah. Okay, so that is for the G1 checkpoint. Okay, I would like you to read more on this G1 checkpoint on page 150. Okay, so that you can understand what is happening dekat G1 checkpoint ni. Macam mana dia exercise the DNA integrity, macam mana dia determine the nutrient availability, and also how they evaluate the growth signal. Please read on that because that is for your knowledge. Walaupun tak ada dalam syllabus for this semester, but please read on that so that you know what is happening dekat G1 punya checkpoint. Okay? So, saya dok sebut apoptosis. So, what is apoptosis? So, apoptosis is the programmed cell death. So, kalau you tengok, refer to the photo here, see, these are normal cells. And then it comes to the fragmenting of the nucleus. So the nucleus will become fragmented. So the cell rounds up and the nucleus collapse. Okay, nucleus dah tak ada, and then the chromatin pula condense and the nucleus fragment. So bila dah the nucleus fragment ni, dah jadi macam luka. So there's blisters everywhere. So blistering of the plasma membrane. So these are called blebs. Okay, tengok ni. These are normal cells. These are the damaged cells. Okay. So, apa jadi lepas ni? So, the cell fragments contain DNA fragments. So, lepas ni dia akan engulfed by the white blood cell or also other cells. Okay. So, apoptosis are caused by the enzyme called caspase. Okay, this is the enzyme responsible for apoptosis. So, mitosis and apoptosis are opposing forces. Mitosis increase the cell number. Apoptosis terbalik, dia decrease the cell number. Okay? So, apoptosis and cell division. So, cell division and apoptosis occur throughout our lifetime to maintain the body's homeostasis. Okay, we keep on dividing. Contohnya sel kita kita terkena cut ke, kita kena apa. So cell division will happen to repair the cells and also to bagi jadi dia baik balik. Okay. Lagi satu contohnya macam dalam embryo, masa kita baby, dalam masa development of the development, masa human development tu, kan ada all this blastula dan everything. Masa embryo stage, okay, tangan kita actually dia jadi macam wet. Dia belum jadi macam tangan kita betul-betul ni lagi. Dia link, dia macam ada wet. Okay, so we need apoptosis to happen supaya dia akan jadi tangan kita macam biasa. So apoptosis will occur during embryonic stage tu untuk maintain, jadikan dia kita as a human form. Okay, contohnya dalam embryo, the web feet and also the tail kita kat belakang tu. Okay, tail tu akan go off due to apoptosis. Okay. However, not all apoptosis are good and not all apoptosis are bad, as we say, tapi it does do its job. Okay, so an abnormal cell that could become cancerous or virus infected cell, they die through apoptosis. Okay, kalau itulah kalau yang cancer ni, kalau dia tak go through apoptosis, itu yang jadi dia semakin merebak. Okay, so the reason actually sebenarnya the cancerous cell yang spread daripada contohnya daripada liver, dia dah pergi ke sebelah, dia pergi ke kidney ke, dia dah pergi tu, is because the apoptosis rate dia dah slow. Maksudnya the checkpoint dia dah tak function fully. Dia function tapi dia tak dapat cope with the amount of rate the rate of the cancer cells. Okay, so that's how cancer can happen. Say because the apoptosis jadi tu dah tak dapat keep up dah dengan sebab cancer cells replicate very fast. Right? So cells harbor caspase enzymes that are normally kept in check by inhibitors. It can be unleashed, caspase enzyme ni apoptosis kan. It can be unleashed by internal or external signals. Okay, so if you read through on page 149, so we have the signaling protein, which is the P15. P53. P, if you refer here, in mammalian cells, the signaling protein P53 stops the cell cycle at G1 point when DNA is damaged. Okay, P stands for protein and 53 represents the molecular weight. First, P53 ni, dia akan attempt to initiate DNA repair. But the rising level of P53 can bring about the apoptosis terus. Kalau dia terlalu banyak tak boleh nak repair, as I said, it will go through apoptosis. So kalau, if successful, the cycle will continue to mitosis. Cuma kalau successful, it could still lead to cancer. Kalau apoptosis, dia terus no cancer risk. Okay? It could still continue to repair ni, tapi there's still a chance for it for cancer to develop. That's why kalau bukan kalau boleh, we cannot control that thing. It's in our body, so we cannot control apoptosis and everything. So if katakanlah the checkpoints are working fully function, semua tu, and apoptosis, and then the rate of cancer punya cells is not malignant yet, so it can be avoided by apoptosis. Okay. All right. So next subtopic we go on into 9.3, which is mitosis and cytokinesis. Okay. So number of chromosomes. How many what is the total number of chromosomes for human? Does anybody know? Ada tak?
23.
23 pairs, right? So total number is 46. Betul, 23 but 23 pairs. Haploid dia, 23 pairs. So bila jadi diploid is 46. Okay, total number of chromosome for humans is 46. Sebab 23 daripada mama, 23 daripada your papa. So 23 and 23 is 46. So when I say diploid cells and also haploid cells, you have to know diploid refers to somatic cells. Okay, and haploid refers to gametes. Okay, right. So that's for diploid and haploid. Okay, these are some examples of diploid chromosome numbers. Okay, dalam yeast, the total chromosome number is 32. Dalam garden pea, 14 saja. Okay, dalam potato, 48. Dalam fern, 1320. Banyak dia punya chromosome number. Fruit fly, 8. Sikit sangat dia punya chromosome number, 8 only. So human, 46 and goldfish have 94. Ni tak payah hafal. You don't have to hafal this. Okay. Okay. Next is the mitosis and cytokinesis. Cuba tengok this GIF here. This summarizes everything that we're going to learn. So from prophase, central stem, spindle fibers, so prometaphase and a metaphase, a line, and then anaphase. After anaphase, we have the telophase where the spindle fibers dissolve. And then lastly is the cytokinesis. Okay, that's the cytokinesis. Okay, so eukaryotic cell division involves mitosis, nuclear division, and cytokinesis, division of the cytoplasm. So during mitosis, sister chromatids are separated and distributed to the daughter cells. So before mitosis begins, so this is the S phase, interphase tu kan. Chromatin condense, okay, they coil into distinctly visible chromosome. So each species has a characteristic chromosome number, which is especially unique to that specific species. Okay, so mitosis and cytokinesis overall steps involves interphase. So we've done that, and we'll go on into prophase. is when the chromosomes or the DNA replicate. And then we have the prometaphase. The nucleus dissolves, mitotic spindles, microtubules attaches to the centromere. And then we have the metaphase where the chromosomes align at the center of the cells. And then next we have the anaphase, which is the mitotic spindle pulls the sister chromatids to the opposite pole. And then we have the telophase. And lastly, we have the cytokinesis. So we'll go into detail in each of these phases. Okay, interphase balik. Okay, during interphase, a cell prepares for cell division. So at the end of the S phase, it consists of two identical double helical DNA molecules or called the sister chromatid. They are attached together at the centromere. So this the single point here is the centromere. Jangan ada yang label nanti sebagai central. This is not central. This is centromere. Okay. So during mitosis, the centromeres holding the sister chromatids together separate, and sister chromatids separate to become the daughter chromosome. Yeah, sister chromatid, and then it will go to mitosis to become the daughter chromosome. So daughter chromosomes of each type distribute to the opposite daughter nuclei. Okay. So comes back to the centrosome balik. Okay, just outside the nucleus is the centrosome. Okay, kat sini. Yeah. Okay, centrosome replicates in the S phase of the interphase. So there are two centrosome before mitosis begin. Dua. Okay, dua. So where does it go? So in animals, the centrosome contains central. Okay, central dia akan extend the microtubules, dia akan jadi mitotic spindle, or we call it as the spindle fibers. So each fiber is composed of a cylindrical bundle of microtubules. So bila microtubules ni assemble, dia ada subunit. Subunit ni we call it as the tubulin. So bila dia assemble, the join, so this is when dia jadi tegak. Okay, all of this akan jadi tegak. So bila the tubulin subunits in disassemble pula, they form the mitotic spindle fibers which allows the cell to change the shape of the cell division. Okay, so that one you have to know the microtubules punya function. Okay, spindle fibers, you have to know what the terms used bila nak guna centrioles, bila nak guna spindle fibers, bila nak guna apa ni, mitotic spindle ke, meiotic spindle ke, okay, all that you have to differentiate. Okay. Okay, so back to the centrosome. Okay, centrosome, kalau animals we call it as the centrioles, kan. Okay, kalau awal ni dalam G1 phase, which is the exit or early, dia mula-mula dia macam ni, G1 phase dia. So masa S phase, dia ada duplication and the elongation. So the central will become like this. Okay, kalau G2 phase, kalau you tengok dia dah jadi macam ni, compare dengan this is G1, so this is G2. Okay, dia jadi dua, dia tak combine dah pun, jadi dua. So at the early part of the mitosis, dia jadi dua. Okay, satu will go to the north pole, satu will go to the south pole. Central, so dia akan form the spindle fibers. Okay, next phase is the prophase. So prophase, I'm sure all of you are very familiar with all of these phases of mitosis already. So go through it briefly saja. So prophase, chromatin condense, and then chromosome distinguishable with microscope. Now each chromosome has two sister chromatids held together at the centromere. So nucleolus disappear, nuclear envelope fragments, and the spindle begins to assemble. So two centrosomes move away from each other. This is when I say it is satu pergi ke north, satu pergi ke south. Okay. You can also watch a YouTube video here. I've inserted the link, okay, on Profis. The next, after prophase, we have the prometaphase. Okay, this is the preparation for sister chromatic separation are evident in the prometaphase. So centromere of each chromosome develops two kinetochores. Okay, prometaphase ni dia dah attach, okay, to the kinetochores. Cuma it is not aligned. Kalau you tengok dia punya chromosomes are still not aligned. So dia align fully masa metaphase, not during prometaphase. Prometaphase is only attached to, okay. So these connect the sister chromatids to opposite poles of the mother cell. So dalam metaphase pula, okay, this is when the M checkpoint ada. Okay, so metaphase ni the chromosomes pulled around by the cuticle fibers. They are forced to align across equatorial plane of the cell. So this is the equatorial plane of the cell. We call it as the metaphase plate. Okay, this is the plane through which the mother cell will be divided. Okay, so M checkpoint delays the start of the anaphase until kinetochores are attached properly. Kalau tak attached properly ke apa, it won't go through into anaphase. Okay, that is the function of the M checkpoint. They check the spindle fibers assembly. Spindle fibers assembly. Okay, okay, next. Kalau dah okay, semua dah lepas all the M checkpoint, kita akan go through the anaphase. The anaphase centromere dissolve, so they release the sister chromatic. So this sister chromatic separate at the centromere. So dia dah berpisah. One go to the north pole and the other go to the south pole. They are pulled to the opposite poles along the kinetochore fibers. The poles move further apart. So anaphase is the quickest phase in all the phases, okay, of the cell cycle. Dia paling cepat to happen. Right, next, after anaphase, what do we have? We have the telophase. So telophase ni, spindle disappears, and then the new nuclear envelopes form around the daughter chromosomes, and now there are two clusters of daughter chromosomes. So clusters are incipient. What does incipient mean? Incipients begins to develop. Okay, the daughter nuclei. Baru nak start tapi belum break apart. Okay. So the nuclear envelopes then form around the two incipient daughter nuclei, and each daughter nucleus receives one chromosome on each type. So division of the cytoplasm requires cytokinesis. So the chromosomes become diffuse chromatin again bila dah happen the cytokinesis tu. Dia dah ada cleavage furrow ataupun ada plate, then dia akan go through the cytokinesis. Okay, recap again. Mesti you all dah apa ni? Dah pro lah mitosis ni kan. So always know ataupun understand what is happening in each of the stages. Prophase jadi apa? Early prophase jadi apa? Prophase jadi apa? Prometaphase jadi apa? Metaphase, anaphase, telophase. Please read through in your medal textbook. Semua ada, okay? On page 154 and 155. Okay? Okay. Cytokinesis in animal cell. Okay. Cytokinesis dalam animal cell, it involves cleavage furrow. Cleavage furrow ni, they appear between the daughter nuclei. Okay, tengah-tengah ni. Cleavage furrow atau we call it as the contractile ring. They form kat sini. Okay. So formed by a contractile ring of actin filament. Okay, contohnya dua, it's like you pulling a drawstring punya bag tu. You macam ikat dia and pull it apart. So it eventually pinches the mother cell into two. Okay, dua, tarik. Okay, that is cleavage furrow. Kalau cytokinesis in plant cell is different. Dia tak ada cleavage furrow. Sebab kenapa dia tak boleh? Sebab dia ada cell wall. That's why dia tak boleh go through cleavage furrow because the cell walls is rigid. So what does it have? Dia akan ada cell plate. Okay, cell plate formation. dekat sini. So cytokinesis in plant cells begin with the formation of a cell plate. Mana vesicles ni datang? Okay, it is made in the Golgi. Okay, they eventually fuse into jadi one thin vesicles extending across the mother cells. So the membranes of the cell plate becomes the plasma membrane between the daughter cell. Lama-lama dia akan jadi plasma membrane. And then the space between the daughter cells are filled then with the middle lamella. Yang tu bila middle lamella ni dia akan lama-lama jadi primary cell wall. Okay, so jadi dua. So akan ada cell wall juga. Yang tadi bezanya dengan cleavage poro dia akan ada the contractile ring. Dia akan cubit, dia keluar. Okay, yang ni dia tak ada. Yang ni hanya dia form dengan cell plate. Okay. Okay, function of mitosis in animal and plant cell. Okay, important when there is fertilization, egg become embryo repeatedly kan kita duk. Okay, kalau actually bila, okay, what actually differentiate you and when you were 5 years old? Cuba relate. You 5 years old, you macam mana? And you sekarang you macam mana? Beza paling ketara lah. What is it?
Size.
Size. Betul. Size. Does that mean your cell tu jadi besar? Sekarang dulu cell you masa you 5 tahun, besar ni je. Tiba-tiba sekarang dah jadi besar ni. Is it like that? Does it work that way?
No.
No. How does it happen?
Our
Growth.
cells increase.
Yes, your cell increase. That is why cell division happen. Lagi banyak cells. Bukannya sebab your cell grow sampai jadi besar ni. Satu ni kan, dalam your tangan tu sampai besar ni. Tak, it's not like that. Okay? It is because your cells have gone through many process of cell division. That is why you are your size now. Okay? Right, so that is the development. Lagi satu, it can also bila ada cut atau heal or broken bone man, it also involves cell division. Okay, stem cell. Okay, we nearly at the end of our lecture already, right? Stem cell. So remember that some of the somatic cells kan dia akan go through the G0 phase. Dia akan how do we say, dia tak replicate, dia tak go through cell division dah. Dia akan remain dalam the G0 phase. Tapi all these cells usually ada stem cell. Okay, stem cell ni it retains the ability to divide. So contohnya macam red bone marrow, kita kan stem cell. They were able to divide to produce various type of blood cell. So that is why scientists semua suka kita simpan kita punya stem cell. Tapi we are not that exposed to this yet. So that is why kita punya apa ni, medical punya ni is a bit how to say, bukan ketinggalan, macam not as advanced as the Western punya countries. Okay, because diorang during birth ke apa, they tend to sign acknowledgement ke apa, okay, stem cell diorang boleh simpan. The stem cell, kalau let's say katakanlah one day you terpatah tangan ke, you need transfusion ke apa, so they will get that from your stem cells. Okay, tapi dekat Malaysia it is still bukan perkara yang normal lagi lah. Not is here ada dah, cuma it's not as norm lagi. Okay, so by these stem cells they can do therapeutic cloning to produce human tissues that can begin with either adult stem cell, ada dua jenis, adult stem cell ataupun embryonic stem cell. Okay, so embryonic stem cells can be used for reproductive cloning, the production of a new individual. Yang ni selalunya diorang akan buat macam sheep dolly tu kan. All that's are reproductive cloning. Okay, so stem cells can replicate itself or it can be changed into many cell types. Okay, so I would like to go through a bit, just briefly on cell cancer. Okay, abnormal growth of cell is called a tumor. Tumor ni ada dua. It can be benign. Benign are not cancerous. Normal tumor lah kalau kita selalu tanya kalau orang tu tumor cancer ke tak? Tak kan? Okay, kalau tak is benign. Okay, kalau cancerous it is malignant. Okay, benign tumors are not cancerous because they are encapsulated. Okay, here, dia still macam dalam kapsul tu. Dia tak terspread dekat luar. Dia tak ada any restriction kalau malignant punya tumor. Kalau benign, it's not cancerous. Okay? Bila dia encapsulated macam ni, dia do not invade the neighboring tissue atau spread. Okay, dia ada inhibitor dia punya tu kan. Kalau yang ni tak ada, it can spread everywhere. They are not encapsulated kalau malignant punya tumor. They are readily to invade neighboring tissue. That's how cancer spreads so fast. Okay, and it results from mutation of gene regulating the cell cycle. Sama ada checkpoint dia tak function betul-betul ke, ataupun dia punya phase, dia G1 S phase and also G2 phase ada problem or anything. So those are the results. The cancer cells are the result of those mistake ataupun ada misjunction ke apa, disjunction anything. Those are the reason cancer will occur. Sometimes like I said, the normal punya, kalau medical doctors explain is because apoptosis tu, it doesn't fully function betul-betul dah. Dia tak expose the damaged cells. Dia tak dispose of the damaged cells dah. Okay? So what are the differences between cancer cells and normal cells? Cancer cells are non-differentiated cells. Dia tak specific. It can spread. Dia merata, dia suka, dia pergi. Dia boleh pergi ke your hand, dia boleh pergi ke shoulder, dia pergi ke organ lain. From the heart spread to the liver, to the lungs. Okay. Kalau normal cell kita differentiated cell. We are specific cell kita. Okay. And then abnormal nuclei. Kalau cancer cells, kalau normal cells, we have normal nuclei. Cancer cells do not undergo apoptosis. Okay, kalau normal cell kita still undergo apoptosis. Cancer cells no contact inhibition, maksudnya tepi ni. Dia tak ada, dia spread je, dia keluar. Okay, kalau normal cell dia ada contact inhibition. Cancer cell dia disorganized and also multi-layered. Kalau normal cells is only one organized layer. And cancer cells undergo metastasis, maksudnya dia boleh jump. Dia boleh spread quickly. Kalau normal cell, kita remain in the original tissue saja. Okay? So, origin of cancer. What you have to know, dia actually our maintenance of our growth and our tissue cells, it depends on the balance of dua tadi tu. The signals, whether the external and the internal signal. Okay? It promotes and inhibits the cell division. So two types of genes may be mutated when balance is upset, which may cause cancer. So what you have to know are dua ni, oncogene and also tumor suppressor gene. Okay, oncogene, okay, kalau you tengok refer to this here, this is the normal cell cycle. Proto-oncogene serve as a gas. You nak push forward kan? And then tumor suppressor gene, dia macam pull you back sikit. So it is balanced. This is the normal cell cycle. Kalau cancer cell, proto and oncogene ni boleh mutate and it may become oncogene. So lagi laju. Too much gas here. So these are how cancer cells develop. Dia lagi laju. Kalau contohnya lagi satu that can happen is the inactive of the tumor suppressor gene. Bila dia inactive, tak ada break lah. Laju juga lagi. So ini laju, ni pun laju. That's how mutations can happen leading to cancer. Kalau kita ada proto-oncogene and we have also our tumor suppressor gene which serves as the brake, we will have a normal cell cycle. Right? Okay, what are the causes of cancer? So it is not definite. Tak ada orang cakap, oh sebab makan Maggi daripada umur saya 5 tahun saya dah makan Maggi dah sampai saya besar. Tak. We cannot say that is definite. Okay. But it is caused by poor diet and nutrition, intake of tobacco. Siapa yang smoke tu kalau boleh kita stop lah, okay. Okay, and then infection, in case ada infection apa-apa, exposure to UV ke, X-rays gitu, and also kalau kita duduk kat bahagian atau rumah kita dekat dengan industrial ni, environmental pollution. Okay, so all of these factors will cause the genetic changes. Don't worry, we will learn more on the mutation ataupun the what causes the genetic changes in the DNA tu nanti masa expression punya topik. Okay, this is just as a heads up for you. I would just like to share on cancer. Okay, so contohnya kat sini kan normal cell division, cell damage, no repair, okay, cell suicide ataupun apoptosis will happen kalau normal cell division. So ni kalau cancer cell division, apoptosis does not happen. So first mutation dah jadi macam ni, second mutation, third mutation, fourth later lama-lama jadi uncontrolled. Banyak. Okay, so that's how cancer cells mutate. Okay, so that is all from me for today for mitosis. Thank you very much for your attention. And I have a question here. So just to see if you are concentrating or not in class. So apa ni? The question is which is not true. Not true, okay? Of the cell cycle. A, the cell cycle is controlled by internal external signals. B, cyclin is a signaling molecule that increases and decreases as the cycle continues. C, the DNA damage can stop the cell cycle at the G1 checkpoint. D, apoptosis occurs frequently during the cell cycle. Siapa-siapa yang nak volunteer to answer? Anybody?
D, Miss.
D, yes, you are correct, meaning you have paid attention during my class.